So, good evening, respected judges. This is Sriyansh Guru, and today I'm going to speak about my project, which is plasmid-based biosensing platforms for biomedical and environmental applications. So, first of all, from the industrial soils, we'll have a bacteria which was formerly known as Ralstonia metallidiurans. Currently, it is known as Ralstonia eutropa. It is a strain of CH34, and it is known to survive in high wastage areas and areas with heavy metal concentrations. It has two plasmids, so this through the process of soil dilution would be brought to us. And then moving on, we'll have to actually culture this bacteria with a vector in which we'll be later putting our plasmid on. That would be E. coli. E. coli is chosen as it has no antimicrobial resistance. So both of these cells have to be cultured first. Moving on with the culture, we need to isolate the plasmid of the Ralstonia eutropa CH34 bacteria, which is basically a prokaryotic bacteria, and it is obligate to toxic environments, which means it can only survive in toxic environments, and in normal environments generally does not survive, but can also modify itself or can mutate itself to the environments. Now, first of all, we need to isolate the plasmid of it through the process of isolation of plasmids. Now, we need to gel electrophoresis. Through gel electrophoresis, we need to study the sequence of the plasmid. After studying, we need to PCR. PCR is a method for which we can get many copies of this plasmid. Now there is a method. Now we need to design a restriction endonuclease enzyme by studying the sequence of this plasmid, which will allow us to extract more plasmids from the bacteria Ralstonia metallidorin CH34. Due to which now we'll have multiple copies of bacterial plasmid. Now, this through a very specific process called heat shock, where the vector or E. coli bacteria is kept upon an ice plate and then immediately put into boiling water for 45 to 55 seconds, where also the plasmid is inserted. Due to the calcium or buffer of calcium is added, due to which E. coli develops pores and it accepts the plasmid, and then it is put back onto ice surface so that the pores close up and E. coli accepts the plasmid. Now, the reason we've chosen E. coli is because it has no AMR, it would happily accept the plasmid and would show less mutations. Compared to Ralstonia metallidians, if it is directly put outside, it is vulnerable to mutations. Now we need to culture this new GMO. Now, just for the conditions, just to see if our test has actually worked out or not, we need to. There is something called 16S rRNA sequence testing, where it is genetic sequences needs to be matched with the library. We need to make a library, and then there is a process called blast test, where our achieved sequence is matched with NCBI's general sequence, just to see if we have got the same bacteria as plasmid or not. Now, since this bacteria is obligate to toxic environments, which means it will only survive in toxic environments, and if it is put out to normal environments, it will die. So, based on how long it survives in a normal environment, we build a toxicology scale, due to which we can compare on the toxicity level and the presence of metals, due to which we can basically calculate on how long should we be there or not. Is that area harmful for us or not? This can be used for discovering new areas, metals, mining, and further it can also have medical applications to check if the presence of metals in further human bodies. Thank you for the project. This is Deansh Guru signing off.